that genes were performed first, and as I was in Boston, in Andrzej Królewski group, we performed the first linkage analysis. So John Rogus made some kind of calculation and found that the best model to perform the linkage analysis would be to collect sibling concordant for type 1 diabetes, but discordant for diabetic nephropathy. It means both of them have type 1 diabetes, one of them develop end-stage renal disease or has proteinuria, and the second one is free of increased albumin excretion after at least 15 years of diabetes. So we check first only 3 regions, the regions which contain the genes for renin-angiotensin system, and we found on chromosome 3, unexpected, that this region which contained the gene for angiotensin 2 type 1 receptor is linked in this population to diabetic nephropathy. So another study parallel to our study with the same model was performed in Sweden. They collected the same sibling with the same model, both with type 1 diabetes, one of them with diabetic nephropathy, the second one without. And they performed first the whole genome linkage analysis, and they found the strongest signal exactly on chromosome 3. So in the Boston group, the whole genome linkage study was performed, and somehow the chromosome 3 was not more the strongest one, but it was somehow strange, 2 groups, 2 models, and they find the same region. So here's a summary of different linkage studies performed. So the first one is this from Jocelyn with the highest lot score. The second one is from Scandinavia. Other studies reached lower lot score for this chromosome 3. So they went further. The group from Scandinavia, from Sweden, went further, and they checked the whole region. They genotype above 3,000 polymorphisms in this region, and they found that one of the polymorphisms, this RS 1866813, is associated with diabetic nephropathy in Finnish population. It was somehow not confirmed in the population from British Islands and from Iceland. So we checked it in our Polish population, and we do not find any association of this variant with diabetic nephropathy in Polish population. From one of the review paper, I know that this variant was also checked in Boston in their population, and it was also not confirmed. So somehow, even we have some data, some proofs that chromosome 3, this region, is linked to diabetic nephropathy, finding the variant, the real one, which will explain at least the positive linkage finding, has not been done yet. So to genome-wide association, with the time the possibility appeared to perform the whole genome-wide association, and the first study was performed also in the group of Andrzej Królewski in Boston. And so we do not see here in the genome-wide association study any signal on chromosome 3, but the strongest one somehow appeared on the chromosome 13. So in the same year, another study, genome-wide association, was performed from Ireland, and they also found some signal on chromosome 13. So this chromosome 13 somehow overlaps in those 2 studies. So on chromosome 13, there were several genetic markers with positive association with diabetic nephropathy. So in the study from Ireland, one of them on the chromosome 3 overlaps, so what is very, very interesting. So the group from Japan tried to repeat the results with this polymorphism, and there were 4 research groups which tried to prove it in their populations, and they were not able to prove it first when they combine all the data together from all the studies they got from one of them a positive finding. So this chromosome 13 seems to be interesting. It probably does not explain the whole heritability of diabetic nephropathy. It's probably, with this odds ratio of 1.26, will not have much indication in clinical practice, but probably will be a way to find a new pathway, pathophysiological pathway to explain diabetic nephropathy and to find probably the treatment, pharmacological treatment. So also the chromosome 13, one polymorphism was confirmed once again in Joslin Diabetes Center in their population to be associated with diabetic nephropathy, and this is right now the strongest signal. So jumping to the summary, there are some promising loci associated with diabetic nephropathy on chromosome 3 and chromosome 13q. As I said, the data has to be evaluated further. It's not ready right now to introduce it into clinical practice, and probably in the future we will get something from this. Right now, still the research is going on. Thank you very much.
We have time for a couple questions for Dart. So what do you think is the potential for finding a region in the genome that may overlap risk of retinopathy and nephropathy, given that they so often tend to occur together?
Yeah, somehow the studies, some of them were performed parallel, some of them were performed separately. With progression of diabetic nephropathy, the retinopathy progressed parallel, so somehow appearance of diabetic nephropathy speed up the progression of retinopathy. And we don't know right now, is it like the same genetic effect, or is it the effect that something is going on in the whole body when the kidney is damaged. The same story is with cardiovascular diseases. So, there are some populations where the risk of cardiovascular disease is low, like Pima Indian, and when they develop diabetic nephropathy and stage renal disease, the risk increased dramatically. It's hard to say. So the data are not so promising then for monogenic forms or even for type 1 and type 2 diabetes.
Other questions? Yes, Thomas.
Are you aware of studies on nephropathy regressors, or are we talking about 2 different populations to study them?
Regressors who regress from diabetic nephropathy. From like clinical observation, from the first stages, it is possible to stop it or to regress it. I do not see much promise, at least in clinical practice, to regress from very advanced stages of diabetic nephropathy. And there are some studies, Mike Maurer, where they transplanted the pancreas and they saw the biopsies some kind of regression of diabetic nephropathy. But as I said, in the first stages it's still possible. In the advanced stages it's almost impossible. Okay,
if there are no further questions, thank you, Dart.